Hi everyone, it's Mr. Sinti, and today I'd like to talk to you about the formation of male and female gametophytes in angiosperms. And gametophytes need to be formed in order for fertilization to take place. And so you might be familiar with the flower, and so today's conversation is going to be all about the flower, and it's going to focus in on the anther, and it's going to focus in on the ovule, because it's the ovule where the female gametophyte is being produced and the anther is where the male is. And then I'm going to take you from gametophyte formation to fertilization, which happens inside the ovary. So pretty cool. We're going to start off with male and female gametophytes, and we're going to end up with a seed. So let's get right into that conversation. It's a very good one. And so male and female gametophytes develop within the anthers and the ovules, respectively, or ovules. And so the anther sometimes is referred to as a pollen sac or a sporangia. How do you like that? House of spores. And so let's get into that. And the female gametophyte develops within the ovule, which is located inside the ovary. Oftentimes there's more than one ovule. There's only one shown here in this picture. And so the diploid plant, if you remember this from a previous discussion, is called the sporophyte. The sporophyte produces the gametophytes, and the gametophytes are haploid, and the sporophyte is diploid. And then this part right here may not make much sense right out of the gate, but it will at the end, sort of retrospectively. But what's happening is spores divide through mitosis to produce the gametophytes. And so you're going to start off with these spores and then undergo meiosis and then mitosis to produce the gametophytes. And then the gametophytes themselves produce the sperm and the egg, which results in fertilization. So let's get into that. So if you were to take a look at an anther and you were to slice it in half right here, and you look at the pollen sac and the sporangium, inside there's thousands and thousands and thousands of little tiny spores. And they're very tiny. They're called microsporocytes. So microsporocytes. And they're diploid, as it turns out. So this is 2N, if I were to label this. 2N, microsporocyte. So what's going to happen to the microsporocyte is that it's going to undergo meiosis, predictably, because you want to produce gametes. And so what happens is meiosis 1 and meiosis 2, and then what you have here are 4 cells. And these 4 little spores, if you will, within one cytoplasm are called microspores, and each one of those are haploid. Let's put an N right there. So microspores, so meiosis. So if you just took one of them, if this is happening to all 4 of them, but it would get too busy. If you look at just one microspore, each of the 4, what's going to happen is that haploid cell is going to actually divide by mitosis to produce 2 cells. And so let me just sort of, it shows it here, but let me just take that over here. So here's one of the microspores. And so it's going to divide by mitosis, and it's going to produce a cell that's known as the generative cell. Let me just write that there, generative cell. And then another cell, which is adjacent to it, which is going to be called the tube cell. And this cell is going to eventually form the pollen tube, which is going to need to travel down the style. Now these are both haploid. This is haploid, this is haploid, haploid. So it's just haploid dividing through mitosis. And so let me switch colors here. These 2 cells are enclosed in this waterproof, very rigid, thick cell wall, which collectively, this wall right here in these 2 cells is referred to as pollen, right there. So this pollen, like in other words, here's a visual right here. This pollen grain is comprised of 2 cells, and collectively, these 2 cells are called the male gametophyte. The generative cell and the pollen tube cell is the male gametophyte, or pollen is the male gametophyte. Now, what's going to happen is the tube cell is going to form, it's going to germinate a tube down the style once it lands on top of the stigma. And what's going to happen over here, let me just illustrate it, is the generative cell is actually going to have its DNA replicated. Let me go back to this. I don't know if I can go back to this color. It's going to have its DNA replicated. So it's going to actually have two nuclei. And these nuclei, the two nuclei, are both haploid, and they're considered to be the sperm nuclei. The sperm nuclei. And there's going to be two of them. That's going to be important later in this conversation. And so that's when I said before that the male gametophyte makes the sperm. And so there's the male gametophyte making the sperm, which are these two nuclei right there. So that's in the male. And so let's take a look over here on this side on the female. But before I do that, let me just continue this male conversation. I'll come back to the female. So this is what pollen looks like under the scanning electron micrograph. It's got this thick wall. It's each sort of surface is unique to particular species, so that species are reproductively isolated by one another because gametes aren't. In other words, pollen isn't able to fuse with the egg, and so therefore there's isolation. So they're unique that way, and they also resist drying out, so they can live for hundreds of years. So within the sporangia, in other words, within the anther, the pollen sac, are all these microsporocytes, and the microsporocyte is diploid. That undergoes meiosis to produce the 4 microspores, and then those divide through mitosis to eventually become the pollen grain, which is made up of 2 cells. One is the generative cell and one is the pollen tube cell. You can see the 2 nucleus right there. Here's another picture of pollen right there. So the microspore divides by mitosis and makes the 2 cells, the generative cell and the pollen tube cell. So here's a picture of pollen. You can see the 2 nuclei, the generative cell and the pollen tube cell right there. So the generative cell and the pollen tube cell. Now what's going to happen is this pollen, when it's on top of the stigma, if I can draw that, let's do that. So here's the female stigma. So now here we are entering into the female. So here's the pollen tube. So what happens is this nuclei develops into 2 sperm nuclei. So that's what you have here, and then this is the pollen tube nuclei. And so now we're going down the style. We're looking for the ovary and we're looking for the ovules. That's where we're going there. And so I like this picture. So it sort of says, okay, what's this? This is the microsporocyte. This is diploid. What process is this right here? This is meiosis. And then if you looked at one of them right there, the microspore, through mitosis, this is what this is. Mitosis, it becomes 2 cells, the generative cell and the pollen tube cell. So collectively, this structure is pollen. So this is the male gametophyte. Pollen is the male gametophyte. Okay, so the female gametophyte is referred to as the embryo sac, and it's comprised of many cells. It looks like this. It's not just the egg. So let's take a look at that. Inside the ovary, there are these numerous ovules, and inside the ovule, you have something called, just like in the male, but instead of it being a microsporocyte, it's large cell. It's called a mega. Let me write that out. It's called a megasporocyte. Let's see. Megasporocyte. And the megasporocyte is a diploid cell. It's going like that, 2N. And what's it going to do? It's going to undergo meiosis. And if you recall, in female meiosis, you have 2 cells and then you have 4 cells, but one of them gets most of the cytoplasm. And so you end up with 4 cells, but one really large one. So that's meiosis. And this is referred to as a megaspore. A megaspore. This cell right here. And so that megaspore is a haploid cell, megaspore. And then just like in the male, so here's the 4 of them. Notice how one of them is much larger. This is the megaspore right there. Megaspore. Okay, megaspore. And then what's going to happen? So mitosis. Are you ready for this? This cell's going to undergo mitosis, but in the female, mitosis occurs 3 times in a row. So if you have one cell, you're going to have, or one nuclei, you're going to have then 2, and then 4, and then 8. If it happens 3 times, you're going to have 8 nuclei. You're like, wow, this is wild, mitosis. So you have 8 nuclei. There's 2, there's 4, and then there's 8. But check this out. In the megaspore, the cytoplasm hasn't divided. But then, right at the very end of those 3 mitosis, then the cytoplasm divides. So you get 1 cell, 2 cells, 3 cells, 4 cell, 5 cells, 6 cell. And then what happens is there's a cell division that incorporates 2 nuclei inside. So you get 1 cell that's really big that has 2 nuclei, so it totals up 1, 2, 3, 4, 5, 6, 7 cells. 7 cells, but 8 nuclei. Okay. And so what you have there is, let me see if I can illustrate that. You get this cell here. There's 1, 2, and 3. This is inside the ovule. These 3 are referred to as the antipodal cells. And then down here, one of these cells is a special cell, which is going to be the egg. I think you know that the egg is going to need to be fertilized by the sperm nuclei. And then these 2 cells that flank the egg are known as synergids. And then this large cell in the center, right here, it has 2 nuclei. 2 nuclei. So there's 1, 2, 3, 4, 5, 6, 7, 8. 8 nuclei, 7 cells. And then there's an opening right here. It's hard to see it, but there's an opening called the micropyle, right there, the micropyle, and that's where the tube is going to penetrate, right in here inside the ovule. Okay. So the megasporocyte then grows into megaspores, we talked about this, and then eventually it'll divide through mitosis 3 times to produce 7 cells, and those 7 cells are referred to as the embryo sac, and that's the female gametophyte is the embryo sac, so all 7 of those cells. The 3, these 3, 6, and then 7. And so up here, just to review, up here this is a diploid megasporocyte, and then through meiosis it produces 1 surviving megaspore, and then 3 mitotic divisions, and then you have 7 cells. Now this big cell in the center is referred to as an endosperm mother cell. How do you like that? Let me write that down if I can. So this big cell in there, I'm not sure if I can. I'm not sure if I can do that. Maybe it's located here. Well, here's a picture of the embryo sac, and the cell that I'm trying to draw, but it doesn't really want me to draw at the moment, is the endosperm mother cell. Right there. And then here's a diagram showing that. So let's do the female. So in the ovule, there's many ovules involved here. So through meiosis, you're creating the megaspore, and then through mitosis, you're making many embryo sacs, and that's what's inside there. That's the male gametophyte. And then in the male, the microspore produces microsporocyte produces microspores through meiosis, and then mitosis creates the pollen, which then forms the tube and goes down. Okay, and so all of this is, it's great and well, but without pollination. Now, pollination is the transfer. It's how does the male get to the female. So this could be aided by wind, or it could be that animals are involved in this, or some kind of insect that helps the pollen reach from the anther to the sticky stigma. And so before I get into that, let's see if I can, get out of this. I'm not sure if I can get out of this. It's funny, there's always some kind of, I apologize, there's always some kind of technical something here that I have to, let's do force quit. No, maybe he doesn't want me to do force quit. Let's do force quit. There we are. All right, so this is where I wanted to go anyway. So let's take a look here. This is a pretty cool animation on YouTube of what we've been talking about. And so let's take a look here. So what's happening is you could see in this animation, here's the flower, looks like some kind of lily right here. And so we're going to focus in at the base of the style. This is the ovary, and inside the ovary you have these ovules, okay? So that one cell was the megasporocyte, and the megasporocyte is dividing through meiosis to produce 4 cells, but only one of them is going to survive. It's the megaspore. And then the megaspore gets really large, it's haploid. But then it's going to have 3 mitotic divisions. That's 1 that produces 2 nuclei. That's 2 that produces 4 nuclei. And then 1 more will produce 8 nuclei. Look at that. Look how exciting this is. So there's 8 nuclei, and you're like, wow, what's going on now? So the nuclei look like they're migrating a little bit. This is where the opening is to the ovule down here, which is called the micropyle. Then look at this. Cell walls start to form. These are the antipodal cells right over here, which its function is kind of unknown, at least to me, that is. And then these 2 cells flank the egg. Notice how the egg is red in this animation. It's very special. This is the cell that needs to be fertilized to produce the zygote. We think that these cells help to sort of guide the sperm to the nuclei. And then there's this big fella right here, or this big gal, I should say, which is the endosperm mother cell. And so now in the male, so that was the embryo sac, those 7 cells. So in the anther, or sporangia, house of spores, you get all these spores up there. Look at that, isn't that cool? So you get the spore. What's inside the spore? The spore is the male gametophyte. The spore contains 2 cells, the pollen tube cell and the generative cell. Notice the generative cell has 2 sperm nuclei. So this is pollination. Apparently it's guided by the wind, and it's traveling. Oh, look, a sticky stigma. It attaches onto the sticky stigma, and there's where the pollen tube starts doing its thing. It makes a big tube, and it's traveling down the style. Look, you can see right through the style. That's kind of cool. And then the sperm nuclei are traveling all the way down into the ovary, inside the pollen tube. They go through the micropyle. Look at that. The sperm nuclei divide into 2 separate nuclei. And so this is a main event right here. The pollen tube nuclei doesn't matter. One sperm fertilizes the egg and produces the zygote. So now that's diploid. This is going to be the future plant. This is the sporophyte of the future. This second nuclei fertilizes, let me pause it, fertilizes the endosperm mother cell, which creates a triploid cell, 3N. So fertilization, this is a come away message here, fertilization in the ovule is actually double fertilization, because you're fertilizing not only the egg, but you're fertilizing the endosperm mother cell. And so you're like, what is the endosperm mother cell? What is this? Well, notice how it's huge. And so what it does is it sort of protects the egg, and it acts as a nutrient storage. So it provides lots of nutrients for the growing embryo to be. So it acts as a mother that way. It provides nutrients. So the endosperm mother cell provides nutrients for the growing embryo. And so ultimately, this will form a seed inside. So this is double fertilization. Let me just pause that there, and I have this one to show as well. This one's pretty cool. And so this one actually has some sound. Let me go sound on this. Let's try that.
Where the young plants are likely to survive. At each step, there's an emphasis on protecting the offspring. This activity reviews some of the most important steps. Events in seed formation begin with pollination, in which wind or animals bring pollen to the flower.
Now, pollen's the male gametophyte.
The growth of pollen tubes cause the flower to discard its colorful parts, petals and stamens. After pollen arrives, the bright petals are a hazard. They attract animals that might eat the delicate young fruit. We've enlarged the ovary and sliced it open. An ovary contains one or more ovules that will become seeds if they are fertilized. One ovule is shown here, with the front removed to reveal its parts. The ovule is attached to the ovary wall by a stalk that provides nutrients. Starting from a pollen grain at the stigma, which is not shown, a pollen tube grows down through the carpel to the ovule. It carries 2 sperm that enter the ovule. One sperm fuses with the egg cell to make a zygote, which is the first cell of the embryo. The other sperm fuses with 2 nuclei in the central cell, making a triploid nucleus. This triploid cell will grow and divide to make a food storing tissue, the endosperm.
What's really interesting about this, let me pause it for a second. What's interesting is, what's the point of the double fertilization? I believe that it would be a gross waste of energy if the endosperm wasn't providing nutrients for the fertilized egg. So I think in that way it assures that not every ovule is going to get fertilized. So not every ovule is going to produce a seed, but the ones that do get fertilized are going to have endosperm, and therefore it's more efficient.
Double fertilization converts the large central cell from diploid to triploid, while the egg becomes a diploid zygote. The surrounding integuments remain diploid as they were before fertilization.
Okay, so that was a cool animation. Let me see if I can go back over here to the notes. I had to force quit it a second ago, so we'll see if everything's fine. Let's see how things are working out on that. So let's close that down, and we'll close. Let's see. Maybe it'll remember where we were. Yeah, that's good. It's nice when it works, especially when you're recording. Okay, so what I wanted to say about that, and then we were talking about pollination. What I wanted to say now is, like, when you talk about fertilization, usually you encourage it and you want fertilization to take place, and even self-pollination, in other words, on the same plant, can produce variation. The truth told is there's more of a likelihood that you'll have variation in the next generation of seeds if you're cross-pollinating. So there's some interesting mechanisms that I just want to kick around for a couple minutes about what prevents self-fertilization. There's some barriers that prevent that really contribute to genetic diversity by assuring the sperm and egg come from different plants. So now one of them, you already know, is that when a plant is dioecious, in other words, dioecious meaning that they're either carpellate or staminate. In other words, they are not bisexual. So you're assuring that you're going to have cross-pollination for sure. But even in plants that are bisexual, now you could have monoecious. In other words, sometimes the pollen doesn't reach the male and female flowers, but then sometimes there's some other mechanisms that are at play there. One of them is even in a bisexual plant, sometimes, check this out, that the anther and the carpel mature at different times. So if they mature at different times, they couldn't possibly fertilize. And then sometimes the way that which they're structured is sort of arranged in a way that it's unlikely that an animal pollinator could sort of transfer it on the same flower because of the location of the anther. So you get all kinds of interesting mechanisms happening there. And even I find this to be most interesting, that there's sort of a self-incompatibility. So in other words, like how our immune system works as an animal is that our white blood cells recognize foreign cells. So this is kind of an interesting thing, like the plant can recognize self, so it's self-incompatibility. So it will reject its own pollen. And so ultimately, double fertilization gives rise to the gives rise to the zygote, which will become the embryo, and it gives rise to the endosperm, which is this nutrient triploid cell, which supplies energy for the embryo. And so what happens is, ultimately, we've been through this. It's the male gametophyte uniting with the female gametophyte, and here's the endosperm mother cell right in the center, which is providing the nutrients. And so ultimately, double fertilization is fertilizing both the egg cell and the endosperm. So you get a diploid and you get a triploid. Whoa, pretty interesting. So that's why it's called double fertilization. And so double fertilization ensures that the endosperm will, I mentioned this, will only develop where the egg has been fertilized. So this prevents squandering of nutrients. And so that's kind of efficient of the plant. And then finally, what's kind of fascinating is that, you think we understood everything about fertilization, but it wasn't until recently that we've been able to really isolate these small structures in a test tube, under a petri dish, under the microscope, and we're able to study that it turns out that there's an increase in cytoplasmic calcium following fertilization. It's very similar in males. In males, that creates a barrier that prevents more sperm from fertilizing, so you don't have an abnormal chromosome number. And so similarly, plants try to block polyspermy, in other words, the multiple sperm nuclei from fertilizing their egg. And so it has to do with calcium, and it has to do with producing like a thick cell wall which prevents other sperm from penetrating. And so this is probably way more than you've ever wanted to know or never thought was possible. I mean, I don't want it to detract, I just want to say this in conclusion, I don't want it to detract from the beauty that is a flower. I mean, even I look at a flower and I think, gee, that's great. I love the colors and the fragrance and what it symbolizes and how we share it with our friends as a symbol of affection. It's a great and a wonderful thing. They make the world bright and beautiful. I know they produce seeds and they're functional. And we went into all this cellular gametophyte conversation, formation of gametophytes. But in the end, flowers are beautiful and blossoms are a cool thing. And so I hope you enjoyed our conversation of the formation of male and female gametophytes.